Terrific. Can you hear me? All right. I realize I'm the only thing holding you up from lunch and the announcement. I'd like to thank Aubrey for inviting me to this wonderful conference because aging is still the biggest risk factor for Alzheimer's disease. There are 35 million cases in the world today, and another 200 million people are walking around today who will get this disease over the next 30 years. So solving this one's like curing cancer 10 times over. Okay, let's start at the beginning. In 1901, a German psychiatrist working at the Frankfurt Asylum had this patient, August Dieter. So Alois Alzheimer was her doctor. She was 51 years old and severely demented, confused, disoriented, paranoid. She couldn't make any new memories. So he followed her for 5 years until she died, and then he took her brain to Emil Kraepellin's lab in Munich, where they discovered the pathology that underlied her condition. So he wrote up a paper for that, a small paper. It's Über eine eigenartige Erkrankung der Hirnrinde, on a peculiar disease of the cerebral cortex. Okay? It wasn't until about a year later that Kraepellin wrote his textbook and mentioned the case, and he referred to it as Alzheimer's disease, and so that's why it's called that. He didn't name it after himself. Okay, so the most obvious pathological feature on a gross level of Alzheimer's disease is on the right here. It's severe atrophy or wasting of the cerebral cortex. It's due to the death of billions of neurons. Okay? If you take a section of that and you cut it up, you find the two pathological features that they identified in Kraepellin's lab. So on the top here are plaques, and those are tiny waxy deposits of amyloid beta protein and some other things, but mostly amyloid beta. Okay? And tangles. Those are these little Eiffel Tower shaped things here. These are the insoluble remnants of the cytoskeleton of neurons that have died. So they're a very good marker for where the cell death has happened in the brain. But neurofibrillary tangles are something that sick neurons make. So it doesn't really tell us why they're sick. It's just that there were sick neurons there. But amyloid has been the center and a preoccupation in Alzheimer's disease research for 25 years. But unfortunately, it has not gone so well. It's been an unbroken string of failed clinical trials, okay, costing about $20 to $30 billion in private and public money, okay? And the bloodletting continues to this day. In just February of this year, 2 companies dropped out of it, okay? So 1,800 clinical trials have been done on something related to Alzheimer's disease. And do you know how many of those trials have slowed or stopped the progression of Alzheimer's disease? 0. Not a single one. That's what I call systematic error. So, oops, there we go. It's a systematic error. So a few years ago, I began to apply Occam's razor to this, looking for a more simplistic solution. Is there something basic that we're missing? Something that it can account for the age-dependent incidence of the disease? Something that accounts for the higher risk for people who have traumatic head injuries and the near certainty that people with specific mutations will develop the disease at an early age, just like August Dieter. Okay? So I'm going to need to use a little bit of neuroanatomy here, but I'll try to keep it simple. Okay? At the top here, we have the right half of the cerebral cortex, okay? The right is the front and the left is the back, okay? The one below that, since it's just the half, is what it looks like on the medial surface, on the inside. And the bottom one is what it looks like from below. So here we have 3 stages, early, middle, and late stage Alzheimer's disease. Now this is neurofibrillary tangle staining from Brock and Brock, 1991. And if you notice on the left-hand side, this orange here is a specific area where the pathology starts, okay? It's almost like a little campfire. It starts off with a spark, it smolders a bit, and then it explodes. And it goes to other regions of the brain, okay? But it starts here and this is called the medial temporal gyrus. It's got the hippocampus in there and big parts of the olfactory system, which are connected there. So there's something peculiar about this area. No other animal gets this disease. And in humans, it starts in this one specific area. And this is actually a different part of the cerebral cortex. The yellow part up here is neocortex, a 6-layered neocortex. But these areas down here, they're allocortex or other cortex, 3-layer and 5-layer cortex. They're a more ancient part of the brain. So what is it about this part of the brain that seeds Alzheimer's pathology, seeds the deposition of amyloid beta in the interstitial spaces of this region of the brain? Okay, I think it comes back to the evolutionary origin. So here is an alligator brain. And the blue area is where the olfactory system is. It used to be a third of the forebrain. Very important. Okay? And if we look for other animals, we can see in mammals, most mammals, it's very large because it's important for smell, okay? Which is important for survival and breeding and evolutionary fitness, okay? But in humans, notice how small it is. It's just a tiny thing, and it sits right below the prefrontal cortex, where executive function and abstract reasoning happen. So it's almost as if there was a turf war for space, and the olfactory system lost. So we think very well, but we can't smell very well. This is my dog Rex, actually. So when I take him for a walk, he smells as if it's an 80-inch plasma, ultra high definition TV, and I smell like it's a little 1950s black and white with the fuzzy lines and stuff. Okay, 70, 100 times better than average human. Okay, now the olfactory system is where the disease starts, where Alzheimer's disease starts. And these are the olfactory bulbs here, the little orange areas there. So what is it about this area that seeds Alzheimer's pathology? Okay, I think it might have to do with the clearance of interstitial spaces. So here's an allegory. Think of a forest, okay? There's a little stream in the forest and leaves fall down from the trees. If they fall on the ground, they sit there and they rot. If they fall into the stream, it carries them away, all right? Later in the summer, the stream starts to dry up and the leaves keep falling in. Eventually, it hits a point where the water can't carry them away, and then they form mats or plaques, right? So I think it has to do with the clearance of the tissue. Now, in most of the body, this is done this way. So capillaries have these tiny holes or fenestrations, or sometimes there's gap between the cells. And blood plasma comes out of these holes and enters the interstitial compartment where it becomes interstitial fluid. It passes slowly through there, picks up insoluble macromolecules, maybe pieces of apoptotic cells, and other debris. And it carries it along to the lymphatic vessels. They take it up, lymph nodes eventually taking it back to the bloodstream. And of course, any cancer cells are going to go along this route, and that's why they'll wind up in the lymph nodes. Okay? But in the brain, this system doesn't exist because the blood-brain barrier prevents any formation of fenestrations or gaps between the endothelial cells. So the brain does it a different way. It has these large cavities, fluid-filled cavities, or ventricles in the middle, shown here in blue. And those have choroid plexus, and they produce cerebrospinal fluid, about ½ liter a day per person. Okay? It percolates through the tissue, through the cortex here, through all the spaces, and along the blood vessels, and it works its way up to the surface, to the subarachnoid spaces on the surface. Okay? From there, it's absorbed or resorbed along the way, but some of it passes all the way down to the spinal cord, where you can get a lumbar puncture or a spinal tap to sample the CSF. Now, remember I said the olfactory system used to be a very big, important part of the forebrain. Well, it sits here, okay, and this is the hippocampus. So this is the area where the disease starts. So CSF comes into there, and it works its way up to the surface of the medial temporal gyrus, but then it goes along the olfactory route, okay? It follows along this route here to the olfactory bulb, okay? It goes a different way, okay? Let's look at it this way. Here's the medial temporal gyrus, okay? The fluid comes in, goes on the surface, goes toward the front, and there's a rudimentary cone here. right? The lateral olfactory stria is this loose fiber bundle, and the CSF passes through these little channels in there along past this area, and for the neurobiologists, the basal forebrains in there, cholinergic neurons, just inside there. And here to the olfactory bulb. And then what happens to the fluid? Well, here's the skull, okay? The front is up here, the back is down there. There are two little depressions or pockets, olfactory fossa, in the front of the skull. And the olfactory bulbs sit in there, and the CSF drains through, okay? So if we look at this figure down here, we can see the olfactory bulbs on top, a little gap between here, although it's a very small gap. Here's the cribriform plate, and here's the nasal mucosa, okay? So here's the olfactory receptors. They're sending signals up these processes up to the olfactory bulb about the odors that are received. But there's gaps in there, and the CSF, the metabolite-laden CSF, makes its way through into the nasal mucosa, where there's plenty of lymphatic vessels. Anybody who's ever had a nasal vaccine, that's why. There are a lot of lymphatic vessels in the nasal mucosa. Okay? So what happens with this? Because this is a natural choke point for the clearance of this fluid from the brain region where the disease starts. Okay? So here is a 26-year-old. It's just a photograph of a dried skull. Okay? Here's a CT image of that. You can see there's some thick bone here, but not very much. We look at an 80-year-old here, and you can see there's a lot more bone deposition in that area. Look at this structure up here. This is actually a piece of bone that sticks up. I'll show you more in a minute. It's called the crista galli. Notice how much bigger it is here in the older person than the younger person. We have continuing bone deposition in this area. And one of the effects of that is it's closing off those holes or those apertures, reducing the ability to clear the CSF. Okay? So we've been, before I go on, I just wanted to show you, here's Kirk Douglas and Michael Douglas. So they have very similar faces, okay? The cribriform plate's about an inch in there, right? And so if you looked at his cribriform plate, it would look more like that, and Michael's, of course, would look less ossified. Okay, so here's a CT scan, and this is the eye socket. This is from the front. Here's the eye socket here. The brain's up here, and here, this is the olfactory fossa. So the cribriform plate's on the bottom here, and this bony structure is this thing I pointed out here, the crista galli. Okay? And then notice it goes down the middle, right here, all the way it connects up with the bone in the middle of your nose, the nasal septum. Okay? So here's a cribriform plate here. This is the back wall, and it's sort of a slant there, so it comes in, it slants in, right? And here's some apertures there. Okay. So what we've been doing is doing a high-resolution micro-CT imaging of about 70 human cribriform plates. These are taken from control subjects of different ages and Alzheimer's patients. And this is a 26-year-old. I actually have a 3D print because we do a lot of that. So maybe if you want to see it later, you can come up and have a look. Okay, it looks way better in person. So here's the back up here. You can see the holes, plenty of holes. There's a little thick bone here, and lots of apertures. Swiss cheese, practically. All right? If we look at a 70-year-old control, we see that there's a bit of a bony veil. It's come down a little bit, and there's not so many apertures in here, right? There's a little bit of bone deposition. Now, if we go a little further, and we look at an Alzheimer's patient, notice here, this big bony veil is covering pretty much all the apertures at the very back of the olfactory fossa, okay? So for reference, this here is where a little blood vessel comes out. There's another one on that side. That would be this area here. Right? So you can see how much more bone deposition we have. And also, see there's a big bony area here? It's because this cribriform plate is uneven. So the crista galli is in the middle. If you break your nose, right, it's going to deflect that, and this thin bone will get broken. And just like a kid who falls off his bike, when it heals, there's going to be extra bone at the site of injury. So what's happened here is we can tell this has been deviated. This is actually elevated, and it's thicker bone on that side. So it seems that if you injure the cribriform plate, it's going to affect this system. So here's those, and I don't know how well this one's going to show up. This is a different different Alzheimer's patient. You can see the big channels are different here, but the big veil is very similar. Okay, so we're talking about how much capacity there is to drain the CSF. Seems to be different in Alzheimer's patients. Here, I also have a print of one of those too. You can come and see later. Okay, so what we're doing is we're doing these high-resolution CT scans, and we've been doing them on cadavers, but we want to start doing them on humans because we want to be able to relate what we see in the high-resolution samples with what we'll see in a lower-resolution clinical sample. Okay, so we do that, we get our scans, and here's some of the hardworking interns at the company here, there, and there. What they're doing is segmenting the files. So these are very large files. I have to go through and say that's bone and that's other tissues. Okay, and what we do is produce these models, like the one I just showed you, which is this one, the young one. And then if we take this one aperture, we blow it up like this, you can see it's got all of these dots. And each dot has an XYZ coordinate. So we get a big file that has thousands upon thousands of these XYZ coordinates. And that's how we're matching them up, okay, is using computers. Okay? So if we do this, what we should be able to do is theoretically look at someone's scan, and when we're all finished with our database, say, what's your theoretical CSF flow capacity, you know? And if we can do that, then we can use CT scans to say, well, this person obviously has a lot of occlusion here. This person's fine. And that can come in very handy for mild cognitive impairment. So that's the pre-dementia state. And only about 10% of those patients progress onto Alzheimer's disease. But the big question's always been, which ones? Well, if we can do the scans and say, well, this one and that one and that one, it's going to be a big help. Okay? It could also be part of an annual wellness visit for seniors. Maybe they get a scan every 2 years or 5 years, and we can actually watch the progression of the ossification and calculate how long it's going to be before they hit the critical threshold. So the bone is kind of only part of the story, because the other part is the soft tissue. So this here is the highest resolution ever done of magnetic resonance imaging, MRI, of a human cribriform plate. It's 11.7 Teslas. And what we see here, the soft tissue is blue, and the bone is this fiery color. We just happen to pick this color. But you can see where the apertures are, but that's not quite good enough because there's so many apertures, there's a couple dozen apertures, and some of them have blood vessels, some of them have nerves, some of them probably have the channels, but we need to know which are which. So we came up with another method. We figured out a way to enhance the staining so that we could use CT and resolve bone and nerve and other tissues in the micro-CT images. And then we, as the interns were showing, we go in and digitize it. So here you can see the bone, right? Here's the nerve. And these blue things here, these are the conduits, fluid-filled conduits where the CSF is actually flowing through. So we can use this with our database to figure out how much CSF flow capacity there is, okay? And here's another image. Here's the nerves here, and you can see the CSF flow channels here. And here's looking at a cribriform plate from above. Here's the crista galli at the front here. Here's the back, okay? Here are the nerves in orange and the flow channels in blue. If we remove the bone, you can see them all here, right? Now the yellow, that's something else that's a little interesting, and nobody's ever seen that before. That's here. These are actually channels within the bone. So they're CSF conduits, and they're connected to these drainage channels. It's almost like an equilibration of the CSF flow mechanism, okay? And it's interesting, these CSF conduits, here's a control, here's another control, not as good as that. We start looking at the Alzheimer's patients, and they're almost completely gone. This is actually in the very back, so the cribriform plate's up here. And so we're losing these conduits. We're also getting ossification. So there's really something going on here with the CSF flow dynamics, okay? And the proof of concept we're doing for this is we're blocking the cribriform plate in ferrets. Turns out mice and rats, for a number of reasons, are probably the worst model you can pick for Alzheimer's disease. It's ironic because the NIH has been very draconian about you must use mice. But anyway, I can explain why later. So anyway, here's our team of human neurosurgeons who brag now that they're the greatest ferret neurosurgeons in the world. So they go in, and this guy has actually had surgery a couple months before. So they go in the top of the skull, go into the nasal cavity, make a little window, and basically peel off the tissue off the cribriform plate and put bone cement on there to block it up. And we want to see after 6 months, do we get plaques and tangles? Hopefully that'd be great. But in the meantime, we're doing behavior studies. Here's me and our technician Zoe, because I want to know the result. So here's the maze that they run through. And so we're assessing them every 2 weeks for behavior. And when we finish this study, we'll put it all together. Okay? So it's one thing to tell somebody, okay, we've done a scan and you're going to get Alzheimer's disease in 5 years or 7 years, or you have a very high risk because of your cribriform plate. It's another thing to be able to do something about it. Okay? So this is Greek mythology, a water nymph, Arethusa. Okay? And she attracted the attention of a river god, and he couldn't get away from him. But Artemis saved her by turning her into a hidden underground stream. Okay? So that's what we want to do. We want to make a hidden channel, or an under the tissue channel, to drain the fluid from this area. Okay? We call it Arethusa. Okay, here's a simple diagram. Basically, we go up through the nose, cribriform plate's there at the very top of the nasal cavity, and we put in the device. This is a very old version of the device. We have a much newer, better one now. But it's in the patent, which is publicly available. So what we feel we'll do is put the shunt in, maybe in people who have mild cognitive impairment, okay? And that should actually reverse their mild cognitive impairment, because the MCI in those patients is probably happening because the A beta has become oligomeric, which is toxic to synapses. And so the synapses aren't working as well. And this is even before they form plaques. So if we restore the flow, we should be able to reduce the level of oligomeric A beta and hopefully prevent the disease from progressing to AD. So that's our plan. I just told you that. So we think it's a minimally invasive, although neurosurgeons do it because we are kind of puncturing into part of the brain, and we have to be very careful about that. We think it should be pretty straightforward. And the trial, since we're doing MCI, isn't going to be the 10 or 20-year trial for an Alzheimer's study. It's only going to be a couple years, because we should see the effects in a matter of weeks to months. And then hopefully 5 years later, they don't progress to AD. Okay. So in summary, I talked about the cribriform plate as the final outlet for the brain region where the disease starts. It's a natural choke point. Okay? We saw age-dependent changes in the cribriform plate, and the patients show more occlusion, obviously. And then the CSF channels, so the fluidics. In terms of looking at these flow channels, it's just a lot of work, because these files are enormous and it takes weeks and weeks to go through one and identify all the channels and all the nerves. But you know, we're making good progress on that. And then what we really want to do is use all of our information to make a diagnostic algorithm that we can get an appropriate CT from anyone and figure out where they are on the spectrum. And then obviously treat MCI. I'd like to make some acknowledgments. Dave Gabel from the Methuselah Foundation back there, he gave me the first funding for Leucadia. He said, when I talked at a SENS conference a couple of years ago in San Francisco, he came up and said, this is really great stuff. I'd like to fund it, but you should start a company. And it was a great idea, because I really appreciate that. And Sergio Ruiz from the M Fund, he's helped with some follow-on funding for the company. Obviously at Leucadia, we have Zoe's our technician and our band of bioengineering interns. And then we have the neurosurgeons at Arrowhead actually did the surgery on the ferrets, and Desert Regional Hospital, Javed Siddiqui, and at USC, our high-resolution imaging is done there by Tia, and Randy Wolcher is the pathologist in Portland who sent me most of the cribriform plates. And oh, that's something else. Maybe somebody will ask a question and I'll pull it up.